Thanks so much for joining me for part 2 of Parker Biology's Cell Communication. In this edition, we're going to be looking at how we know what we know about cell communication, along with the actual mechanisms of how cells talk to each other. Take a few minutes to go ahead and read about Sutherland and his epinephrine experiments, and think about what the results show and how anything described up above relates to your prior knowledge of carbohydrates and proteins. Pause the video now, please. The main idea of this passage is that epinephrine is a hormone, and during times of extreme mental or physical stress, it's released, which is going to stimulate the breakdown of glycogen into glucose. And the enzyme that helps facilitate this is known as glycogen phosphorylase. Why does it make sense that if you're under extreme stress that you would want to break a glycogen into glucose? Well, remember, this is our complex storage carbohydrate. So it would make sense if you need a lot of energy for fight or flight that you would break that storage compound into usable units of glucose that are more readily available to go into cellular respiration and yield ATP. So from here, Sutherland wanted to see how exactly epinephrine was involved in stimulating glycogen phosphorylase to catalyze that breakdown of glycogen into glucose. When he put the epinephrine in with just the substrate and the enzyme, the breakdown didn't occur. Glycogen was not broken into glucose. He found that epinephrine could only activate this enzymatic reaction if it was added to a solution with intact cells. Interestingly enough, this shows that epinephrine does not directly interact with glycogen phosphorylase. Instead, since intact cells are required, it seems like the cell membrane is actually necessary for activation. If we think back to the Part 1 video where we looked at cell signaling, this reminds me of the idea of a receptor being needed to interact with that ligand or that signaling molecule. So perhaps that receptor is located on the cell membrane. Now, up in these passages, it really should be raising some red flags that remind you of information from earlier in the year. So any of the earlier information about carbs and proteins that rang a bell? Well, I'm hoping that term glycogen rang a bell. That idea that glycogen is simply a polymer of glucose. Remember, that's how animals store up their carbohydrates. It's sort of the animal equivalent of plants' starch. Also, when we think about proteins, hopefully you recognized an ending up there, the ASE, that clued you in to the fact that glycogen phosphorylase is an enzyme. Also, this should remind us of prior knowledge about proteins, that specific proteins called enzymes catalyze reactions. The further we get in the year, the more comforting it is when we find information that reminds us of things we've already learned. So Sutherland's experiments with epinephrine show us that intact cells were required, therefore the membrane was needed for activation. And here we're going to look at a bit of an overview of this whole process of cell signaling. So before we mentioned signaling molecules, the ligands, sending a message, that the message has to be received, and that there are a variety of events that take place inside the cell. Ultimately, cells talk to each other to bring about some sort of response. Maybe they want to turn on a gene. Maybe they need to manufacture a protein. Maybe they need to activate an enzyme. So even though there are different types of responses that can occur, the pathways are very similar. As seen here, we can divide the entire event into 4 substeps: signaling, reception, transduction, and response. Take a minute using those Roman numerals and find those events up in the picture, please. Pause the video here. You should recognize that signaling will be associated with the signal molecule or the ligand. Reception requires a properly shaped receptor protein, shown here. Transduction refers to the series of events that occur within the cell, a series of changes that ultimately bring about the cellular response. The message is sent, the message is received, changes occur, and the cell does something about the message it got. Just to match these terms with actual textbook definitions, we can say that in the reception phase, the signal binds to a cellular protein. During transduction, that binding of the ligand is going to lead to changes in the receptor that trigger a series of changes along this pathway. And finally, what's the cell going to do about it? In response, there'll be a specific cellular activity. Take a few minutes with the Sutherland epinephrine experiment and think about how that experiment involves each of these 3 steps. Describe which parts of the experiment fit with each stage. Pause the video now, please. In reception, we should see the chemical signal bind to the cell. And in Sutherland's epinephrine experiment, this occurred when the epinephrine bound to the receptor. Next should be transduction. So here we should see a series of changes, and that's exactly what happens with epinephrine. There are a series of changes that occur to activate glycogen phosphorylase. The true goal of epinephrine's message coming to this cell was to stimulate the breakdown of glycogen into glucose, to have that readily available energy source. So that'll be the cellular response. Again, even though different cells talk about different things, this theme of reception, transduction, and response is incredibly common across lots of different types of cell communication pathways. Here we saw epinephrine binding, a series of changes to activate the enzyme, and finally, the enzyme catalyzing the breakdown of glycogen into glucose. Next, we're going to look in more detail at reception. There are 2 scenarios here. We've got nerve growth factor, which is water soluble, and testosterone, which is a steroidal hormone. So I want you to think about each of these different signaling molecules and decide where you think the receptor should be. Should it be intracellular or located inside the cell? Or should these receptors be located within the plasma membrane? Think about the properties of substances that move easily through the plasma membrane and properties that would imply maybe it needs to stay on the membrane instead. Pause the video here and discuss with your neighbor which would be found inside and which would be found on the plasma membrane. It's important to think back to our discussion of the plasma membrane to remember who moves easily through it and who has a more difficult time. As we look at the phospholipid bilayer, remember it's comprised of our phospholipids. We've got our hydrophilic heads and our hydrophobic tails. So keep in mind with this, small hydrophobic materials move more easily through. They need to interact with that highly hydrophobic tail region. So small hydrophobic materials or small nonpolar materials do move more easily. So what does that tell us about each of these signaling molecules? Let's look first at the nerve growth factor. Remember, it did tell us that this is a water-soluble molecule, which implies that it is in fact hydrophilic. Do water-soluble hydrophilic things move easily through? No, they interact well with those heads, but not with that dense tail region. So the idea that this is hydrophilic tells us it's not going to be able to diffuse through the membrane, and therefore its receptor must be located on the membrane, not on the interior of the cell. It would have too hard a time getting there. What about testosterone, this steroidal hormone? What do we remember about steroids? Keep in mind, they're lipids. So if they're lipids, they are nonpolar, which means that this receptor could be intracellular. It could be located on the inside of the cell because this signaling molecule would move easily through the membrane. Now, whether the receptor is found on the membrane or inside the cell, the receptor does need to be complementary in shape to the signaling molecule. This ensures that only certain cells respond to certain messages. These receptors are absolutely vital in biological systems. Anytime something malfunctions with a receptor, we can see disease results. So by better understanding these receptors, we can also help improve treatment of disease. Thanks so much for joining me for part 2 of cell communication, where we examined the epinephrine discovery experiment and look more at receptors. Join me for part 3 to understand 3 different types of receptors and to look at more details of transduction and response.